Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we will be talking about proximity labeling for drug discovery with Ciaran Seath today. Good morning, Ciaran.
Hey, good morning.
Yeah, good to have you on the show. And now, without further ado, I want to introduce our guest, Ciaran. So Ciaran completed his PhD in chemistry at the University of Strathclyde in 2017 under the supervision of Alan Watson, and he studied chemoselective transition metal catalysis. During his PhD work, he also spent time in the labs of Tom Snadden at Indiana University, working on the total synthesis of complex natural products, and with Glenn Burley on the development of novel click chemistries for nucleoside bioconjugation. He then moved to Emory University as a postdoctoral researcher, working in the laboratory of Nathan Dewey, developing novel photoreductive photoredox methodologies, specifically working on new synthetic methods to access alkylated heterocycles and complex fluorinated scaffolds. In 2019, Ciaran moved to Princeton University to work with David McMillan and Tom Muir, where he then explored new photocatalytic methods for proximity labeling to investigate critical aspects of cancer biology. Currently, Ciaran is an assistant professor of chemistry at the Scripps, Florida, in Jupiter, Florida, where his group uses subcellular interactomic techniques to uncover new druggable targets. And Ciaran, I don't know if I can say that already, but you're moving to Cornell University, right?
Yeah. I am. In July 1st, we'll start Cornell.
Amazing. Well, it's an honor to have you on the show here today.
Thank you so much. A great introduction.
Awesome. And yeah, before we get started with the talk, we love to learn a little bit more about our speakers. So, Ciaran, what inspired you to start a career in drug discovery in a drug discovery related field?
So when I was young, you make these choices quite early in the UK when you apply for university. I ended up applying for chemistry with medicinal chemistry as my degree path when I was 16, and that was kind of medic adjacent. I kind of wanted to be a medic, but I'm somewhat squeamish around blood. And this was the closest thing, right? I was like, oh, well, I'll still be in the field, but from a chemistry side. And then ever since then, I did a year at GSK in Philadelphia during my undergraduate degree. And then in my PhD in Alan's lab, while I worked on catalysis, the other half of the lab worked on medicinal chemistry. And that has kind of been mirrored throughout my career. So half of Nate Dewey's lab worked on medicinal chemistry and nuclear receptor targets. And then Macmillan's lab is obviously heavily involved with the pharmaceutical industry. And that's something I've continued in my lab with some method development, some molecular biology, but also small molecule drug discovery.
Yeah, super cool, super cool. Yeah, I'm the same way actually. I also can't see my own blood and other people's blood.
Yeah.
As a chemist, of course, we often think in molecules. So I wonder, and you've worked on many complex natural products and stuff. What is your favorite molecule and why?
Yeah, I had to think about this. I'm not precious about these sorts of things, but I picked a molecule that I taught on recently. So this is a molecule called trapoxin. So this is a cyclic peptide natural products, like fungal natural products, synthesized by Schreiber in 96. The synthesis is fine. It's a very important compound for target ID. It was one of the first examples of that was immobilized to beads and identified the target. So from a chemical biology point, it's quite important. And it also kind of kick-started the field of epigenetics. So this molecule was vital in the discovery of the first epigenetic eraser, so HDACs. So it's a molecule I think that incorporates many of the aspects of my career. So epigenetics, target identification, and small molecule synthesis. And I think it's a great advert for total synthesis, which I think is chronically under appreciated.
Yeah, yeah, makes sense. And macrocyclic peptides certainly celebrate a comeback right now in the drug
Yeah,
discovery industry
yeah.
with
It's covalent as
icotrokinra.
well. Yeah, it's covalent as well. So,
Oh,
it's
wow.
hits a lot of boxes these days.
Yeah, I love that. I love that. It seems like a paradigm-breaking molecule. Definitely, again, something that's celebrating a huge comeback in the industry. Super
Yeah.
cool. Yeah, no, I didn't know about that molecule, but yeah, now I'm going to read more about it. Super cool. What are you most excited about in drug discovery today? You already touched on a couple of different topics, and of course, we're going to hear about proximity labeling today. What's
So
super exciting to you?
I think compared to when I was getting taught medicinal chemistry, like I think the rules don't really matter anymore. Um, and some of the molecules that are getting released in terms of making new protein interactions, blocking protein function with protein interactions, biologics, the incorporation of small molecules with biologics. I think some of these approaches are just unbelievably innovative, and really allow creativity in thinking in a way that maybe classic small molecule drug discovery was more limited by a set of rules that we all get taught as undergraduates. And I feel like now, you can trust that, with good molecules, yeah, they will be effective, regardless if they're huge molecular weight. I mean, I think that's just great for the industry to explore these different modalities.
Yeah, no, I'm totally with you, right? We just had the approval of the first heterobifunctional degrader. People are now looking at antibody degrader conjugates,
Yeah,
and
yeah, yeah.
as you pointed out, Kieran, the possibilities seem to be limitless, and especially with technologies now that your group is developing, pursuing those harder targets with those harder molecules becomes ever more simple. Not
Yeah,
simple,
well,
but
yeah.
easier, right?
Yeah,
Yeah.
it should be, you see these molecules like, the Revolution Medicines RAS molecule. These are really amazing. The way, this is kind of breaking a lot of paradigms, and it's really, super exciting. Covalency and inducing new gluing interactions to turn off proteins. Like this is super cool. Like I love that. I love this stuff, and the molecules are crazy from a synthetic point of view, which I also love. I love to see these. I look at them online, and it's like a challenge for my group. I was like, "Who come up with a route for this?" Like
Amazing.
it's fun.
Love that. So yeah, if you could go back to the beginning of your career, what kind of career advice would you give your younger self? So what are you telling your students today what they should be doing?
Well, I would probably say to my younger self anyway, um, something, Macmillan said a lot is keep the faith. If you work hard and you are dedicated, there's a good outcome is going to be there for you. I think probably PhD students now are quite worried about this with how difficult it is to get jobs in the industry. But I think it's important to remember, like, if you're dedicated, you work super hard, like that will be rewarded. And maybe I would have, I think, I was worried about those things as a student as well, that, never get a faculty position and how hard it was. However, for those who knew me at that age, would also know that I wouldn't have listened anyway. So, yeah.
Yeah, keeping the faith. That's certainly good. I love that.
Yeah.
And yeah, last but not least, what do you most like about Drug Hunter?
Yeah, I think Drug Hunter, in combination with maybe Derek Lowe's blog, is kind of the official, industry magazine now. This is where we go for our news. It's very hard to digest the literature and certainly regulatory information. So having a place, a beautiful website we can go to in the morning and read a really interesting article and learn about cool new compounds is just so valuable in an age when, we no longer get CNEN sent to the departments and stuff like that. This Drug Hunter has become this staple in my morning, and I'm sure many people's.
Amazing. Yeah, cool. Thank you so much, Kieran. Well, and for those people who don't know much about Drug Hunter, I wanted to briefly show the platform and show a couple of things that you can do with it. So, one of the things that we recently established is actually a course. So, of course, we're going to talk about a little bit more of the new modalities here today, but we actually provide, a drug discovery course here that is meant to be for industry scientists, where we are introducing you to drug discovery, talking about drug discovery engine, ligand binding basics, the dose, and also, of course, here as well in the target ID and validation aspect. That's what we're going to hear a little bit more about today, like these interesting chemical biology technologies. We have lectures about target selection, which of course is very important. Of course, we can't make a good compound for a bad target, right? A drug discovery program on a bad target will always fail if you have on-target toxicity and such. So no chemistry can overcome that. So target selection is incredibly important. Target validation as well. The druggable interactome, I'll just show a brief outlook of how this looks like. Of course, we have a huge number of proteins in the human proteome, 20,000 approximately. 3,000 of those are considered ligandable, so druggable. Then you have about 3,000 that are disease associated but hard to drug. We're going to hear a little bit more about c-Myc today. And only about 1,000 are ligandable at the moment with current technologies and are confirmed to be disease relevant. Of course, we're working on bringing more of this bucket into this bucket here, and this is what the lecture today will be about. You can then kind of learn a little bit more about how the human proteome is built. It's actually, it's of course fairly complex, but we're bringing it down so you understand the major classes and how different classes interact. So receptor tyrosine kinases, of course, are those receptors or enzymes? No, they're of course receptors and enzymes, right? So we're in this particular field. We learned something about GPCRs and signaling, ion channels, kinases, nuclear receptors here as well. So definitely check out our drug discovery course if you haven't yet. And there's many, many more features in Drug Hunter. We have a molecule search. Everything is structure searchable as well as text searchable. We do demos for companies almost daily and can tailor our demo to your specific company. Typically, our customers span from single-person biotech founders to the largest pharma companies in the world. And the platform can also support use cases in growing in a growing amount of functions beyond medicinal chemistry. So in chemical biology, disease area biology, DMPK, even competitive intelligence, medical affairs, and business development. So if you're interested in a demo of Drug Hunter, please indicate so after the flash talk or send us an email at info@drughunter.com. And now, without further ado, I'll hand the podium here to Ciaran for his talk on proximity labeling for drug discovery. Super excited for your talk, Ciaran.
Thank you. Okay. So thanks very much for the invite to give this talk today. Yeah, really excited to talk about proximity labeling and photo proximity labeling and how it can be applied towards problems in drug discovery. So when we think about intervention and correction of abnormal cellular states, I think it's very important to remember just how complicated the cell is. So each cell has around 42 million protein molecules, 20,000 unique proteins, and many more RNA and DNA molecules on top of that. And, set up beautifully by Dennis there, where understanding how these interact can lead to new therapeutic opportunities. So you saw those diagrams are shown on the Drug Hunter website. Understanding these pathways is really the basis behind the development of many therapeutics. So the subject of this talk is really to discuss how these can be measured and then exploited in creative ways, I think, to solve problems in drug discovery. So the traditional approach to understand interactions between biomolecules is a technique called co-immunoprecipitation. So here you would take cell lysate and incubate with an antibody for your protein of interest, forms an antigen-antibody complex, which can then be isolated on solid supports using typically beads. This allows you to wash away unbound proteins to get an enriched complex, which can then be analyzed via either Western blotting or chemoproteomics. And this workflow is generally incompatible with maintaining weak interactions. So I like to think about this as an analogy. So you can consider the cell as a bowl of pasta, and the cell is made up of many parts. So if your question is what interacts with the pasta, and you perform a co-IP, that's like taking your fork and having some pasta, and then you analyze this picture and you come to a conclusion, and that the pasta interacts with the tomato sauce. And that influences how we do our downstream biology. And while that's true, it's not the full story. And when you consider this, in this analogy, there's obviously a need to overcome some clear problems that I think work in a cellular context too. So weak interactions are not detected. So if you pick up the pasta, you're really never going to pick up a meatball. Site-specific interactions are not detected. So the pasta and the herb, right? Depending on where you pick up the pasta, you may or may not get some herbs. And the analysis occurs from the sample in a non-native environment. So on a fork, rather than looking at the picture as it is in the bowl. And molecular and chemical biologists have spent really a lot of time over the past 20 years trying to develop techniques, many different techniques to try and really solve these issues. And what I'd like to talk about today is a technique called proximity labeling. And this is an approach that is used to identify interactions between biomolecules in the cellular setting. And briefly, this works through the localization of some activator, a catalyst, or an enzyme to your protein of interest somewhere in the cell. And then you flood the cell with a high concentration of an inert small molecule probe. Upon external stimulus, this small molecule probe is then catalyzed through some process into a reactive intermediate, which can then covalently label nearby biomolecules, which can be enriched and analyzed via chemoproteomics. If you want to perform an experiment like this, there's a lot of choices. And the two probably most well-used methods that account for over 95% of all literature reports of proximity labeling come from the lab of Alice Ting at Stanford. And this is Apex and TurboID. And these use phenols or biotin as the inert probe to generate either phenoxy radicals or activated esters, which can go on and react with nearby proteins. And in many cases, these techniques are incredible and have been used broadly throughout the industry. However, they do have some pluses and some negatives. So while Apex works really fast, so is really fantastic for looking at temporally controlled interactomes. It's been used for GPCR signaling, for example. The stimulus used for this is toxic. It's hydrogen peroxide. It also has a tyrosine selective label and has a relatively long radius. So you generally need to use spatial controls and a relatively challenging procedure. So if you have multiple plates and you're dosing hydrogen peroxide, wash out 30 seconds later, it can introduce some error, although, it's not a huge problem. In the case of BioID and TurboID, these are non-toxic and can be used in vivo by just adding biotin into feed, but they're relatively slow and they're typically considered always on because they're activated by endogenous biotin. And I've put both of these genetically encodable as positives because it's used very readily by a molecular biologist in a cell line. However, if you cannot perform genetic engineering on your sample, so tissue or in organisms, tissue slices, all this sort of stuff, this becomes a real issue. So we can think about a potential solution to some of these issues. And an ideal experiment for proximity labeling that would address some of these issues could involve a stimulus such as light. So you could switch it on and off with perfect control and also spatial control within a single sample. Ideally, it would label rapidly and can be used in complex settings without toxicity. And light, because you can vary the wavelength, should enable, with appropriate chemistries, the use in higher organisms, tissue samples from patients, all of these sort of samples that we may want to study that may be a bit more representative than transfected cell lines. And the first step towards such an ideal system was developed by the µMap lab while I was a postdoc. So Jacob Jerry, now assistant professor at Cornell Weill, and James Oakley at Pfizer, in collaboration with Nii Fadei and Rob Osland, then at the Merck ESC, developed a technique called µMap. And this technique was really designed to help understand the interactomes of key immunoreceptors. And here, a primary antibody directed to your protein of interest was followed by incubation with a secondary that's conjugated to an iridium catalyst. And this iridium catalyst is pretty special because upon incubation with diazine molecules that are conjugated with biotin, this poises the system for activation. So upon irradiation, this iridium catalyst is able to perform a process called dexter energy transfer. And this occurs only within a couple nanometer radius and turns these inert diazine molecules into highly reactive carbines. And these cross-linked with proteins within only a few nanometers of the antibody. So the real importance of this platform can really only be illustrated in comparison to what was existing before this. So the µMap system, when measured on a kind of an artificial cellular membrane, shows a radius of 54 nanometers. And this labels, in this system at least, around 200 to 300 proteins. If you compare this to the EMAR system, which works with activated azides, this has a double the radius and over double the number of proteins are then labeled. Apex, these peroxidase-based approaches, have an even larger radius, approximately 5 times, which then labels kind of almost the whole cellular membrane. And this labeling radius obviously has a huge effect on the resolution. And I think this is illustrated very, very cleanly with volcano plots. So here, this is an experiment performed by James Oakley, where he labeled EGFR on A549 cells. And you can see with a µMap experiment, EGFR is the topmost enriched protein, and you get approximately 10 proteins to follow up for future analysis. If you compare this to a peroxidase type experiment, you get well over 100 proteins. And EGFR, probably most importantly, is not the most enriched interactor. So trying to assign of these proteins the most important or what you want to follow up on becomes a significant challenge for the downstream analysis. But what I think, and I find most exciting about this initial report, is that it's a chemistry-driven discovery. So it's all based on fundamental photophysics. And because chemists are, innately creative people, this has inspired many other research groups that employ other innovative chemical reactions that extend this general concept broadly across the chemical and molecular biology space. And this is a growing field, and I'm really showing here a selection of the group leaders that have contributed with new probe and catalyst designs, innovative ways to deploy these technologies, and then even extending them into the RNA field and lipid-based labeling. And it's really exciting to see the work of all these researchers and how they're pushing these techniques forward, both from a chemistry and a biological, both in both realms, and it's pretty great to see. So today I'm going to focus on really, rather than just understanding a single protein's interactions, how these techniques can be used to address, I think, major challenges that may face colleagues in the pharmaceutical industry. So I'd like to take a couple of recent cases that I think of emblematic examples of how proximity labeling can be used. The first in how to overcome resistance to immunotherapies, and then an approach from my own lab where we show we can use subcellular approaches to try and find solutions to undruggable proteins. So I'll start out with this story from the µMap lab about overcoming resistance to HER2 immunotherapy. And this was a challenge taken up by Steve Knudsen, postdoc in the µMap lab, who's now starting his independent career at University of Michigan this summer. And he took up this kind of major challenge in the context of HER2 positive breast cancers. So patient tumors can be stratified into HER2 positive or HER2 negative, with positive tumors generally being responsive to anti-HER2 immunotherapies such as Herceptin. However, a subset exists, which you can see in the top right-hand quadrant, that are resistant to these therapies despite high levels of HER2. So Steve questioned whether he could use µMap to understand and overcome this resistance. So to do this, he conjugated iridium to anti-HER2 antibodies and then performed µMap across these sensitive and resistant cell lines. And just to summarize a full paper into one slide, really, there's a couple different very interesting things that came out of this. So on the left-hand side, here's a plot of the interactome size of HER2 correlated with a dependency against HER2. So broadly, the size of the interactome correlates very closely with the dependency. So these resistant cell lines just have many more protein interactors. And when you start to look at these interactors, these resistant cell lines contained a distinct cluster of proteins that were not found in the receptive cell lines. This is showed on the right-hand side. And I just want to focus on their data surrounding this one protein, galectin-1. So inhibitors for this protein exist. And if you take these two cell lines and then plot how they respond to different treatments. So in the untreated, the HER2 immunotherapy and the galectin inhibitor GB1211 by itself, very little change in cell viability was observed over the treatment period. However, in both of these cell lines, the combination of galectin inhibition and the HER2 immunotherapy induces sensitivity to, apologies for the text, induces sensitivity to the HER2 immunotherapy. And by Western blotting for each of these proteins, you can start to understand why. So HER2 in combination, HER2 immunotherapy in combination with this galectin inhibitor leads to 71% reduction in HER2 protein levels in these breast cancer cells. And I think these studies, again, go away and read the entirety of the paper online, but highlight the power of this short radius proximity labeling to provide avenues to overcome resistance. So I think this is a general paradigm that can be used broadly across resistant versus sensitive cancers. So the second story I want to highlight is from my own group, where we've used photo proximity labeling to provide approaches for targeting undruggable proteins. I want to tell one story today that's been driven by a graduate student in my lab, one of the second student to join my group. And that's Ryan Million. And he asked the question whether µMap can be used to identify approaches for c-Myc-driven cancers. So again, Dennis highlighted MYC in the introduction, which was great. And MYC is deregulated in greater than 40% of cancers and is, in many cases, the driver of these cancers. Generally, when MYC is overexpressed, it leads to an overactive cell cycle and dysregulated metabolism, and this rapidly leads to oncogenesis. And this is considered one of the classic undruggable proteins, and there's certainly no clinical ways for direct inhibition, although many interactors of c-Myc which regulate its function have been clinically evaluated, such as PLK1, BRD4, WDR5, and MAX, have all reached clinical trials. However, these generally lead to toxicity due to their impact on healthy cells, as MYC is essential in essentially all of the cells in your body. So any interfering with its native function leads to pretty systemic toxicity. So we questioned whether we could uncover and use µMap to uncover druggable effectors of MYC that are cancer specific. So to do this, we set up an experiment in prostate cancer using 3 models. So WPRIM-Y1, so this is a model for healthy prostate cells as a control. LANCAPS, which are an androgen-positive prostate cancer, which we can maybe consider a low risk or readily treatable with AR-targeted therapies. And then PC3s as a model for AR-negative prostate cancer, so a high-risk cancer, which has no current effective therapies, with an overall goal of being able to deregulate MYC in these PC3 cells may provide like a potential therapeutic avenue towards these high-risk cancers. So when you think about performing interactomics in each of these systems on c-Myc, you can kind of consider each of these interactomes and how we think about them. So anything that's found adjacent to c-Myc in WPY1, this healthy cell, can maybe be considered a healthy interaction that we don't want to touch. Anything that's exclusive to LANCAP and PC3s can maybe be considered as a pan-cancer interactome. And then we'll have LANCAP specific and PC3 specific interactions. And we've looked at all of these subsets, but today I want to focus on these proteins that are specific to PC3s. And in this data set, we identified 345 unique interactors in this cell line. And this is a large number. So in order to stratify these, we correlated them all with their DepMap dependencies. So DepMap is a measure of how essential each protein is when knocked out in this PC3 cell line. And we found 20 proteins that are both adjacent to MYC, only in these cells, and also are important for cellular function. And of these, we consider really only one of them to be druggable. And this was this protein called SLK. And this was interesting to me because I really didn't know anything about this kinase. And the literature on it, relative to most kinases, was relatively thin at the time. So we went on to validate these interactions, and we performed co-IP, the technique I mentioned at the start of this talk. We show again that PC3, the c-Myc SLK interaction is unique to PC3 cells. And at this point, we thought probably SLK is only expressed in this one cell line, and that may account for this interaction. It turns out that's not the case. SLK is broadly expressed in all three cell lines, and it turns out through your body. It's a very ubiquitously expressed kinase. However, when we look at these cells under a microscope and stain for SLK, we get a better picture of exactly what's going on. So in the healthy cells and the LANCAP cells, where there's no interaction between the two proteins, SLK is localized primarily in the cytosol. However, in the PC3s, you have this high concentration of nuclear SLK, which is kind of a unique localization only in this one cell line that may account for its interaction with MYC. So to try and understand whether this interaction was really modulating MYC function, we made CRISPR knockout cell lines in all three, but I'm showing just the WPMY1 and PC3s here, knocking out SLK a very minimal effect on WPMY1s, where the MYC expression is very low. However, the SLK knockout in PC3 cells had a dramatic effect on MYC protein levels. And over 7 replicates, this accounted to around 5-fold change. So pretty dramatic loss to almost healthy cell levels. We also looked at MYC mRNA to see if it was driven by gene expression, and we see a 1.5-fold loss. So while this could account for the change in protein levels, so to investigate this, we looked at how SLK knockout alters the stability of c-Myc. So in PC3s, the c-Myc half-life is around 2.5 hours approximately, and in the SLK knockout, this destabilizes the protein and moves its half-life to around about an hour. So this preliminary stage, we could say that the presence of SLK stabilizes MYC at the protein level. And because this protein is ubiquitously expressed, this is invisible to bulk RNA and mass spec studies that have really driven early stage drug discovery for many, many years. So to try and figure out exactly what's going on here at a molecular level, we took both recombinant proteins treated with ATP and tried to observe and understand if SLK was altering MYC function via phosphorylation. And here's just a snapshot of one of the experiments we performed. So taking these two proteins together, adding ATP, and you can see that SLK leads to robust phosphorylation of MYC and autophosphorylation. So we had many questions. We can see that it directly phosphorylates MYC, but where does this occur? How does it influence stability? And MYC stability is generally regulated through this short peptide called the MYC phosphodegron. And Probably the most important residue is T58, and this is phosphorylated by GSK3b, which induces degradation via the proteasome. So to test if SLK was altering this phosphorylation state, we first took GSK3b and µ, and you see robust phosphorylation of T58. If you perform the same experiment and you add in SLK and a 1-to-1 ratio with GSK3b, you see a drop in phosphorylation of this phosphodegradant to about 60%. And then if you pre-incubate SLK with µ, then add GSK3b, you reduce this further to about 45%. And this is specific to SLK. Other kinases don't exert this effect. And SLK itself is unable to phosphorylate this site, also showing its selectivity. So this started to give us clues about how this was functioning. And it appears that SLK phosphorylation, and probably its presence, reduces T58 phosphorylation via GSK3b. So we took these samples and we ran them down the mass spec to try and understand where µ was being phosphorylated by SLK. And we found this one peptide, so µ329 phosphorylation induced by SLK. And the kinase preferences for SLK are known, and it likes to have a lysine or arginine at the +2 position. And this peptide meets this criteria, suggesting it is indeed phosphorylated by SLK. And turns out you can look through the literature. This is a known stabilizing PTM that was discovered by the Magnuson lab in a different context where it's induced by PIM1 and 2 kinase. And this is really exciting to us because these kinases are a more pan-MYC interactor. And this discovery in 2008 led to extensive drug discovery and preclinical studies against PIM1/2. So just to ensure that this site was indeed responsible for altering stability, we made mutations. So we did site-directed mutagenesis of c-Myc, and we made that serine to alanine mutant and the serine to aspartate mutant. And the alanine essentially mimicking the unphosphorylated, which led to a decrease in stability, and the phosphorylated phosphomimic led to an increase in stability, mirroring our hypothesis with SLK. So we also wanted to see if the presence of SLK induces or reduces the interactions between µ and GSK3b. We performed an experiment called proximity ligation. So each of these puncta is an interaction between µ and GSK. And you can see that SLK knockout induces a robust interaction, a more robust interaction between GSK and µ, which is consistent with an increased degradation. So we're pretty confident now that SLK is indeed altering MYC stability, but does that correlate to tumor growth and cancerous phenotypes? So we first looked at PC3 cells and their cell cycle, which is a common way of looking at c-Myc activity. So in WPMY1 cells, you see it's pretty standard cell cycle distribution. But in PC3 cells, they have this large S phase, which is a marker for rapid proliferation. When you knock out SLK, very little happens to the healthy cells. But the PC3 cells, you get this reduced S phase. And this is just change in S phase is MYC dependent. So if you remove SLK but add in MYC, you go back to the kind of more standard PC3 cells cell cycle distribution. And this is mirrored with doubling times. So knocking SLK out reduces doubling time or increases doubling time by about 50%, and has no statistical effect on the healthy cells. Once again, this change in proliferation is primarily based on MYC. So re-adding MYC into these knockout cell lines brings them back to a more wild-type phenotype. So we're pretty confident in a dish that this was having an effect on cancer growth. So we moved this into a xenograft model, and we grafted onto the flank of mice, either the PC3 cells or the SLK knockout. And in line with expectation, the PC3 cells grew robust tumors over the course of 40 days. However, without SLK, these PC3 cells were unable to form tumors. So you can track each of these mice here and see that all of the PC3 cells kind of have an incubation period and then spike with rapid growth. And we only saw one of these SLK knockouts form any detectable size, and it was very, very slight. This tells us that without SLK, these cells are really unable to form tumors. So our last question was, why does this happen? Why do we have this selectivity for PC3 cells? And this is kind of something that bugged us for a long time. And we, again, always go to the literature, and we found that SLK has two splice isoforms, with the big difference being at exon 13. And this is retained in the presence of these splicing modulators, ESRP1, and skipped with RBFOX2 presence. And this long form is really only found in healthy tissues that exhibit rapid regeneration. And this started to trigger some thoughts for us. So we looked for the presence of this exon 13, and we found it only in the PC3 cells. And this exon contains this peptide that can be considered in the literature as a pseudo nuclear localization sequence. So starting to put together the picture here, where exon 13 inclusion with its NLS moves it to the nucleus, where it can start to regulate MYC, and which would also be consistent with the regeneration phenotype observed in skin. So next, to identify if this is really relevant in human cancer, we started to do some bioinformatics. So we looked at the GTEx prostate database and the TCGA, the Cancer Genome Atlas prostate adenocarcinoma database. This is big RNA sequencing databases of real patients with either healthy or cancer. And we looked at the isoform usage, so whether these cancers have a majority of SLK short or SLK long. And in these healthy prostate, you can see in blue, where they predominate with the SLK short, and the converse is observed in cancer. So these prostate adenocarcinomas have a greater than 75% usage of this SLK long. So this is highly enriched in prostate cancer. To correlate this with MYC in a real patient data set, we then took all of these prostate adenocarcinoma RNA sequencing samples and correlated the presence of SLK long, this long isoform that translocates to the nucleus, and correlated this with MYC target genes. So whether the genes that MYC's controlled are up in these samples. And while we see no correlation with the short isoform, we see a very strong correlation with SLK long. So its expression is strongly related to MYC target genes. And this leads us to a model where in non-cancer cells, SLK, or at least cancer cells that don't exhibit these phenotypes, SLK is localized in the cytoplasm, and it's mostly the short form. MYC expression is controlled by a degradation through phosphorylation with GSK3B. However, in these cancers, SLK is spliced into the long form. This then translocates to the nucleus and blocks GSK3B from phosphorylating c-Myc by phosphorylation of serine 329. And this leads to uncontrolled MYC expression and growth-like phenotypes. So this is a novel cancer-driving pathway, a potential strategy in prostate cancer. And I think what's really important for the context of this talk is this is really only accessible via subcellular approaches. So bulk RNA sequencing or proteomics are just not going to capture any differences between these cell lines. And because of this is a previously unappreciated target. So there's a couple ligands, but they also inhibit GSK3B, which obviously confuses any treatment options. So my group is currently actively working on the development of selective ligands for SLK, and that's something we're really excited about in the future. So just to wrap up, I think µMap and other interactomics techniques are moving in a direction from just interactomics, looking at try and understand the biology of a particular protein that's maybe poorly characterized, to becoming a versatile tool to solve challenges in drug discovery with questions like how to overcome resistance and how to tackle these undruggable proteins. And these two papers, both on the bioRxiv around the end of last year, and hopefully we'll see them in journals soon. Just want to finish by thanking my group. This is my group here in front of the Scripps campus, and special thanks to Ryan, who did all the work on the µMap project. And thank you all for listening.
Amazing. Really great talk here. And I learned so much. I mean, it's super cool method and in the application of overcoming that HER2 resistance with a galectin inhibitor. That's super cool. And then, you know, you guys identifying this new target with this methodology SLK is really incredible. I hope the industry is listening.
Not too closely. Give me a few years, you know.
Yeah, makes sense. Makes sense. We do have a lot of audience questions already. I wanted to start with one question about the resolution, actually. So you were talking about those different proximity labeling methods and their resolution.
Yeah.
I want to hear your opinion. Do you think the limit of resolution has been reached with the current photo proximity labeling with the 52 nanometers, or is there any further improvement that you can see that one could achieve?
So the 52 is the length of the antibody plus 0 to 1. So if you perform the same experiment with a biotinylated antibody, it's the same. So the µMap approach is basically tether limited. So it's as close to 0 as you can be, which in certain of these examples is a real strength, and in others is a problem. With the antibody's flexibility gives you that labeling of both sides. But in other approaches where you have less flexibility, it can actually be too short. And it's completely dependent on the problem you want to address. So I think your primary question has a limit of yes. I don't think you can get shorter, but you don't want shorter, would be my comment. I would say what's really exciting, and we've started to see some papers come out from Alice Ting's lab, Jeff Martel's lab, is how to take these other approaches and increase the resolution of those. I think that's, the µMap approach is because it's a small molecule photocatalyst, and that has its strengths for deployment, right? You're not doing any engineering, you're using native cell lines and tissues. There's also a lot of advantages to genetic encodable enzymes, for example, and that they've done some really amazing work recently to try and narrow the radius of those techniques, which was kind of one of the challenges I highlighted, and we all highlighted back in 2019, 2018, that they were just too long. So I think that's a real strength for these, going forward.
Yeah. That makes total sense. Yeah, and another example probably for overcoming resistance. You talked about overcoming HER2 resistance. I guess if you think about breast cancer, there's also CDK4, CDK6 inhibitors, dual inhibitors, where patients often become and develop resistance for them, and now there are new methods in the industry to resensitize those CDK4, CDK6 resistant tumors with CDK2 inhibition, for example. Have you guys looked into these types of things already or not yet?
So we've looked at epigenetic. We haven't looked at those kinases, but we've looked at, for example, HDAC inhibitors and cell lines that are resistant versus sensitive. And we have some data there, but it's complicated. It gets complicated. So some of these stories are quite clean, but there's others where it gets really, really complicated, and it takes a long time to tease out everything. The challenge with any omics project is mechanisms in biology are rarely simple.
Yeah, makes sense. Yeah, let's get to the next audience questions from Hyun-Song Kim. So they are wondering, how can you address the specificity and selectivity of hit compounds identified from µMap labeling studies? Do you have a unique method or approach for that?
Well, so I showed how we looked at in that SLK context. So there are a lot of other orthogonal data sets, either in the literature or generated by us. So the first things we'll do is okay, is this protein highly expressed, and that's why we're capturing it as a hit, or is it important? And that DepMap will tell us that in the context of cancer. Sometimes we have CRISPR screen data or CRISPR-I screen data if we're looking at ligand specific effects. So how if you're looking at the interactome of protein or complex, you can then use CRISPR screens to filter the proteins that really matter in terms of ligand phenotype. So those are good ways to narrow it down. And then we'll get a list based on what we think is most important. We have an interest in drug discovery. So typically I try and look for proteins that are ligandable, right? Maybe there's other proteins which are completely, unligandable, but ultimately that's not going to lead to a solution. But then, yeah, we use this proximity ligation technique a lot, co-immunoprecipitation when it works, microscopy, super resolution microscopy to overlay. These are all really great methods to validate that proteins interact, and then biochemistry, making them recombinant proteins. Once we've shown that once we're pretty confident, we'll get the recombinant proteins made.
Yeah, super cool. Well, maybe along those lines, and more specifically for those PC3 cells, you identified with the volcano plot, I think, a couple 20 plus different proteins. So the audience is wondering, you mentioned only SLK would be druggable from those. Like, what are the criteria you apply for druggability? For example, I think you had CDK1 in there also.
Yeah, but CDK1, CDK1 is going to induce massive toxicity.
Exactly.
There's a human element to that as well. Whereas SLK, at least we would never, while CDK1 is, we would probably find biology. We're never going to initiate a drug discovery campaign on CDK1, like never. Whereas SLK is something that seems underappreciated. It's definitely druggable to kinase. You can be pretty confident with enough work you're going to get a reasonable ligand. Yeah, so that there's other things in there, but if there are huge tox issues, then we're not going to touch them.
Yeah, makes total sense. Next question is from Nicholas. So he is wondering, is there any hypothesis on why the S phase in the SLK-KO plus MYC is bigger than wild type? It was slightly bigger. He's wondering whether or not this is statistically significant.
It is statistically significant, but it's because when we transfect the MYC in, it's higher. It's a higher MYC. So we made we dosed a lentivirus, but it's not perfect one-for-one MYC expression. The MYC expression is higher, and that's also why the doubling time is faster as well. Yeah.
Got it. Johan is wondering whether you're envisioning targeting SLK through covalent or non-covalent mechanism.
We have a lot of ideas.
I bet.
Again, that's what I love about science. You can see from this talk, okay, SLK is nuclear, and it starts doing the bad stuff when it's nuclear. So we have a lot of ideas in the pipeline of, okay, how do we address it in a way that's non-toxic? You would imagine targeting it systemically is going to have issues with sunlight sensitivity in the skin, maybe have some liver toxicity as well. So how do we target it in a specific manner? So we have a lot of ideas around that. I don't think it doesn't take a genius to think about them, but yeah. But we need to make the selective ligand first, and we're getting there. So
super cool, super cool. Yeah, we're definitely looking forward to seeing that and covering that for sure. People in the audience are also wondering about transcription factors. Is proximity labeling a good method to either directly label transcription factors or their binding partners?
Yeah, I mean, that's where my group focuses.
Yeah.
There's a number of reasons that these are important targets for us. So one, they're frequently drivers of cancer or disease in general, almost always undruggable. They're also really challenging for conventional methods, right? So co-IPs of transcription factors, some work, but most of the interactions are scaffolded, right, by DNA, histone PTMs. So kind of a challenging area. Something we found out pretty quickly is that because a lot of these are small and their environment is dense, putting fusion proteins on them is also a huge issue. So adding large fusions, even pretty small fusions, 10, 12 kilodalton fusions, can just completely wreck the phenotypes of these, right? So if you put apex, for example, on some of these proteins, they no longer function. The transcriptomes are very different. And that, again, is great for us because the photochemical techniques, it's just a catalyst. So if you can incorporate these, small molecular weight catalysts, small relative to a protein, then you can retain function and look at these interactomes. So it works for us. I didn't intend this when I started. I was really just cared about the undruggable nature, but for many reasons, it's a pretty good playground.
Thanks. It's cool. I think Brandon had a question as well.
Yeah, great seminar, Ciaran. Thanks very much. Just looking at the model for SLK oncogenesis had me thinking a bit about the hyperactivation lethality space, right? Well, inhibiting the inhibitors of proteins. And had you all thought about just targeting GSK3 beta in these long splice form cells to see if you can increase the mitotic catastrophe there?
Yeah, I mean, that's a good. My guess is that those experiments have probably been done. Not
Yeah,
maybe thinking about this mechanism, but there's so many GSK3B inhibitors out there.
right,
We
right.
should. This is a good suggestion. We should mine that data at least to understand if it works.
Yeah, because I would imagine, I mean, potential for synergy between those two effects as you pursue SLK, and then
Yeah,
no better place to start than known inhibitors. So,
yeah. Oh no, I believe me. After a year of trying to get a selective inhibitor, I don't know how many tens and tens of thousands of dollars. It's just we're still just scratching the surface. So I completely agree. Yeah, we should look at that data. It's definitely out there. Yeah, it's a great
cool.
suggestion.
Yeah, of course. Thanks.
Super cool. Another audience question would be, have you tested multiple µMap antibodies for labeling, and did they produce a consistent data set?
Yes, we have tested. So in this case, we don't actually use an antibody.
Okay.
We use a intine-based approach that I developed as a postdoc. It's a Nature paper with Tom and Dave from 2023, I think it came out. We have tried to use antibodies for µMap, and they do give different data sets. We haven't found an antibody that's really good yet for this. So that this is a great question, right? It actually gets to the heart of the point, because if you could do this with an antibody, you could start doing it in tissue. You could start doing it in very complicated settings, but we use transfection. Yeah, we've not found one yet, and I don't know if we are going to, because we've tried quite a few. Some of them are okay, but they're not as good as what we use here.
Yeah, makes sense. And then, yeah, I guess we're almost at the top of the hour, so maybe one last question to close it out. So there's an audience question on the reactivity of the diazarines. Are there any guidelines of choosing appropriate diazarines for labeling and micromapping?
Yeah, use that one.
Only that one, right? Yeah, with the
Pretty
CF3,
much. I
right?
mean, as long as the core of the diazarin, so the CF3 and then the benzyl amine. So if that's an ester, it doesn't work, or at least it works poorly, because the electronics around that ring dictate the singlet triplet gap, and that dictates its reactivity. So you get a triplet from the energy transfer, and that'll rapidly equilibrate to a singlet with this diazarin because the gap is large. But changing the electronics around the ring, you get far more triplet, which is then less reactive. So use that one would be my advice. Yeah.
Awesome. Yeah, super cool. Well, thank you again, Ciaran, for a very informative and engaging seminar. I really learned a lot about the technology and its applications in different areas. So yeah, thank you so much again for educating us on this technology.
Thank you so much for the invite. It was super fun.
Super cool. Yeah, thanks to the audience as well for the engaging questions and for being here with us today. We will be back next week with Birgit Wilding, and she will be talking about the discovery of zongertinib, which is a selective irreversible HER2 tyrosine kinase inhibitor. So Ciaran set that up perfectly today here. All right. With that, everybody have a wonderful rest of your day and a great weekend. Take care. Bye now.